Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
Good afternoon, everyone. My name is David Heckerman. I'm the Director of eScience at Microsoft Research, and I'd like to welcome you to the panel discussion. The title will be, you can see it there, What Do Scientists Really Need to Facilitate Time to Discovery? And what we really mean by that is, what do scientists really need other than money and grad students to facilitate time to discovery? And we'll be going in this order. I thought we'd spend about half the time hearing 7 or 8 minutes from each of the panelists. Then we'll open it up to you for questions. So without further ado, let me introduce Jeff Dozier from University of California, Santa Barbara.
Great, thanks. Okay, so amongst the challenges we face for the 21st century, perhaps the greatest one is for water, because of the likely consequences that will happen in climate change. And specifically in the Mountain West, where we get water from snow and also from rain, the question is really, how do we trade off between getting reliable, clean water supply and also maintaining an acceptable flood risk? And if you think about the way one would operate a reservoir, those are diametrically opposed to one another. So this is the way we do it, and have been doing it since 1910. We ski through the Sierra Nevada, and we poke these tubes into the snow, and we pull them up, and we weigh them at about 300 locations, and then we use those in a spring runoff forecasting scheme. And these jobs have very low turnover. A more automatic way of doing it that's been prevalent since about the 1970s with many stations in the 1980s is to put a pressure measuring device at the surface and then to estimate the weight of the snow above that. Now the problem with this is, if we look at the data from these snow pillows, if we look at and snow courses and concentrate on the April 1st measurement, we see that all of the red circles on the left-hand diagram are those in which the April 1st snow has decreased over the last 50 years. And what we see on the right-hand circle is indicates areas where we've had earlier runoff over the last 50 years. So there is a changing snowpack. And this is a diagram that I've put together to show this issue of the end of stationarity. So the red circles are the ones where we've seen a decrease greater than 1% per year over the period of record, which in this case is about 70 years. The diamonds are ones where we've not seen much of a change, and the blue circles are ones where we've seen a slight increase. And the two axes are elevation on the left-hand axis and latitude on the horizontal axis. And so what we see is that all the ones where we're seeing this change are at either low elevation or lower latitude. And so what this means is that the snowpack is in fact shifting over this period of record. And therefore the statistical ways that we use to do runoff forecasting are likely not to work in the future. So even though we have this wonderful record, the problem we face is that all of our experiences in the past and all of our decisions are about the future. So what we are going to have to do is a more mechanistic understanding of what's going on. So this is a way of processing satellite imagery from MODIS to in fact estimate the fractional coverage of each of the pixels. And in terms of doing this, this requires, there's about 38 images per day that we have to process, and the snow season typically lasts about 300 days. And so just doing a month of data requires 1,000 images that one has to keep track of. And some of the images are not going to be good because of two things. One is cloud cover, and the other is because of off-angle viewing, which stretches the pixel. And so what we see up in the upper left-hand corner is sort of a raw data cube for a month of data, where the black values indicate essentially data that I don't want to use. And so then what we try to produce is an interpolated and smooth cube, where for every day we try to produce a value of the snow cover for each pixel. And then what we see is in a given year, and these are the months from January through July, you see the snow cover in the top image, and then in the lower row, you see the reflectance of the snow cover, the albedo. And so the other thing that happens as the snow ages is that it becomes a little bit darker. And then we can calculate the net radiation on the snowpack as a function of the incoming solar radiation, the snow-covered area, and then 1 minus that reflectivity. And therefore, we can calculate the amount of radiative energy that is going into the melt. So in terms of what I need to do better, first of all, I think of data as being a cycle, just like almost a publication cycle, where we collect data, we store, search, retrieve, analyze, present, but then what we create is another data set that in fact either we can use or someone else can use. As Paul Newton noted in his talk this morning, we tend to use a lot of packages, a fairly diverse computing environment, including IDL and MATLAB. We use public packages, say that do radiative transfer. We have our own local code, but I think we tend less to program in C and Fortran than we did a decade or two ago. And so what we need to do is be able to get these programs to communicate with one another easily, and then if we want to put these in cloud computing, we have to have this notion of what I call graduated outsourcing, that I sort of move pieces of what I do out to a higher performance facility. So what I want in order to do better is a pretty easy integration of data and quality checks for a variety of data sets, including surface measurements and remote sensing data, an easy integration of tools, and got it, right. And I really want my cloud to look like my laptop or my desktop. That is, I would like to run a trial version of what I'm doing on this machine here and then pretty easily move it off to the cloud. I need a good way of storing and accessing and visualizing multidimensional data. These cubes are big, and we sometimes want to look through them spatially, but we also want to look through them temporally. And so the data have to be organized in a way that does that. I want a nice user interface to typically with a geographical focus to get to the data. And because I'm putting these data products out for other customers, I need to be able to format output for what they want to use. Okay, thank you. Right.
Thanks, Jeff.
Okay.
So next up, we'll have Brian Traynor from the National Institute of Aging. Brian?
Afternoon, ladies and gentlemen. So I'll just warn you ahead of time, I'm Irish, so I tend to ramble and go on far too long. So I'm relying on David here to tell me if I do that. Just to see if we can get my slide up there, if possible. I don't know where it's gone. Oh, yeah, okay. Always a good thing. Trainer, here we go. Right. All right, so you'll be pleased to know I've just got the one slide, but basically what I try to do is to sit down and think about the things that get in the way of my job. I'm a neurogeneticist, formerly a neurologist, but now a neurogeneticist. And the way I look at it is my job is to make DNA hit chip, these SNP chips that we use for doing genome-wide association. So I try to sit down and think of the various things that get in the way. Well, of course, the first thing is you need the DNA samples. And this is particularly a major problem with genome-wide association because you need thousands upon thousands of DNA samples in most cases, not in every case, but in most cases you need thousands upon thousands of DNA samples in order to be able to distinguish these risk variants that give rise to disease. And the availability of those DNA samples is absolutely crucial. Now, what used to happen in the past is that whoever collected them, the samples would be collected and they'd go into their fridge and they'd never see the light of day again. And of course, that was, a little bit disingenuous in terms of patients because they're after donating the samples, so they want them to be used. So there was this sort of locked fridge syndrome that used to exist in DNA, in the genetics world. Now we've actually shifted a little bit away from that into publicly accessible DNA banks. And the classic one is the Coriell DNA bank located up in Camden, New Jersey, whereby you can actually ring up today and say, I want X number of samples, and provided you're willing to pay for them, you can get them the next day. And I think that has actually really represented a paradigm shift. I hate using that word, but it really has been a bit of a paradigm shift in terms of how we do science. Now, there are many other DNA banks around the world, but in many instances they have what's known as scientific access committees, which I otherwise call fences and obstacles, because these scientific committees, they really should be abolished, I think. I think either the DNA is made available to everybody or it's not, because we need this as our building block. Now, I was at the American Society of, moving on to the next point about faster, cheaper sequencing, I was at the American Society of Human Genetics meeting in Philadelphia recently, and one of the things that was really striking was the rate at which sequencing, the cost of sequencing is diminishing and our capacity to sequence. The first genome, sequencing the first genome cost nearly $500,000. The second one cost $100,000, and I think it's rapidly decreasing every time. And they're actually predicting that later next year, we'll be down to about $5,000 per human genome, and the era of $1,000 genome is not far away, maybe 3 or 5 years, and maybe even sooner. Now, that's absolutely marvelous. If we've all seen that movie Gattaca, where everybody's genome gets sequenced on a daily basis, that's marvelous. But the problem is that it carries with it its own set of problems, namely data overload. That there are so many variants. It's estimated that there's up to 11 million variants, SNPs, single nucleotide polymorphisms, in the human genome. And there's probably a lot more that we don't know about. And that means that, okay, you're going to have all of this sequence data, but how are you going to distinguish these common population polymorphisms, or in some cases just rare, non-disease-causing polymorphisms, from what you're really looking for, the disease SNP, or the disease variant? And I think that comes down to our ability to make this data available to the public, similar to the way we do with the DNA samples, in an anonymized fashion. Of course, we've sequenced somebody's DNA. We don't want the whole world knowing who that person is, so it has to be done in an anonymized way. But crunching those numbers is phenomenal, because remember, we've got 3 billion base pairs in everybody's genome, and you have to find some way of adding that all together. One thought I had was that maybe it might be an idea that every time we sequence a human genome, that we are almost required, if not morally, but ethically, to actually put that genome on the web and make it publicly available. One way in which, one weak point, one key point, lynch point in which we could actually attack that would be through the journal editors. They would say, right, we will publish your paper which describes sequencing the genomes of 100 people, but only if you agree to put that data on the web. So I think that's a very important thing. So on one side, you've got the DNA samples, and the other side, you've got the sequence data that comes out of it. Faster computers really is going to be, as I say, a key component to analyzing all of that data. Then there's another little sort of bugbear. I'm about to get up onto my soapbox here a little bit. In the genome file, in the genome world, we publish and tend to have things that are very good, papers that are getting to high impact journals are those which basically purport to find associations, in other words, positive studies. But what happens when 12 months later somebody else does a bigger study involving a greater number of DNA samples and finds exactly the opposite finding, in other words, a negative result? Well, what happens is that that negative result tends to find its way into lower impact journals. But if you think about it, as scientists, what we're looking for, what we're searching for is not positive results. We're searching for the truth. And both negative and positive results should be viewed equally, as equally important in both of that. And I think it's something that should be considered. And David mentioned we're not allowed to mention money, so I'll leave out the last one about encouraging junior investigators. Okay, thank you very much.
Thanks, Brian. Okay, next up is Jeremy Frey from University of Southampton.
Thank you. So I have to make this do something. Where is mine? There we go. Here we go. So first of all, I must apologise for the fact that I don't have much of a voice. I've taken 2 weeks out, 1 week holiday and 1 week to come here, and of course this is the 2 weeks which I have a cold. So any work on how to reduce the likelihood of catching an infection whilst on an aeroplane would seem to be one of the major ways of improving our scientific productivity. So just to begin with the title that I put on here, Science Ajar, I hope that many of you know the little boy's joke about when is a door not a door? When it's ajar. Just in case you weren't quite sure what I meant by ajar. We have interesting things in formats that are required by Southampton University now, and that's why this divider page here, I haven't removed all of the things. I've just put down a few thoughts about the sort of work that I'm involved with in the sort of environment which is the sort of chemistry laboratory. We'll see, I'll call that sort of moderate scale work. And really, I'm after the idea that smart people need smart places to work in, smart and helpful environments. And the digital world does offer significant possibilities to increase quality and efficiency of the research and the sort of collaboration, and make the best use of what is the most limited and valuable resource, which is the people. People are, on the whole, the most expensive resource, and certainly the most difficult ones if they're highly trained and high quality. And one of the issues that I have are the standards. As somebody mentioned this morning, that can be the bugbear of people's lives, the standards. But in the end, the only way that people can readily communicate is to understand what the material means, is to have some standards. And then suitable and usable tools are a requirement. So issue is always about the availability of the information, and I might have some comments later about the issue of anonymized DNA data. But in principle, the data that you want, if it's to be usable, it's got to be accessible, searchable, and available, but available in a context, and the context is one of the biggest problems. How open this data could be, we could have Peter Murray Rust up here and have a discussion about open data. I don't particularly want to go that route, but when you have decided to make your research available, and what it should be possible to do is to readily and easily make the data behind that available. Whether you're required to, morally obliged to, is a one discussion, but the fact that you should be able to is one of the things where the tools are perhaps not as good as they ought to be. And making sure that this context is moved on. So if I pick up some data from somewhere and reuse it and at least maintain a link to where that data was found, then I want that context information to flow forward. Interestingly, in a case, when I was preparing one of these slides and I copied something out of one Microsoft PowerPoint into another one, and I wanted the image to appear in the slide of the other one, of course it knew it was a PowerPoint slide, so it put it into the deck. So I copied it into Word first, where it promptly forgot that it was a PowerPoint slide, and copied it back into PowerPoint, and it worked. So sometimes it's an advantage when the context is lost. I'm sure there's a way of doing that directly, by the way. But other times you want it to have known what it was and keep remembering what it was. Just to take some of the characteristics of the type of environment that I would like to focus on in my part of the discussions. This is not the only work that I do, but the idea of the smaller scale, long tail science. There are lots of chemistry laboratories around the world. They typically have 1 to 20 researchers. If you're a very successful organic chemist, you might have 50 or 100 people working in your lab. If you're a normal physical chemist, you have 3 or 4 people. And we publish concise and important papers, of course, which reference, they may reference extensive local data sets and international data sets. The problems are with the medium scale stuff. So if you do something once off, and you only ever do this experiment once, then you do it as a once-off thing. You analyze your data and you record it. Hopefully you write down the details properly. If you do something, if you're part of a big collaboration and you do things millions of times, then it's worth investing in the infrastructure to do it efficiently and reproducibly and so on. But in the middle, you have this problem that you suddenly discover that you've done this many times over. Each time you're having to reinvent what you're doing, but never did it seem worthwhile to actually overcome the barrier to do something about it, to automate it in many ways. And in our chemistry environments, it's very frequent that you see students repeatedly doing something or reinventing the wheel, so on. And the question is, why do we do that? And why, as the infrastructure is available, not seeming to map to the needs, and we keep doing much the same thing? But when you actually get to dealing with real people in the labs here, in these small groups, small collaborations, how do you actually persuade them to do something right? Because the general thesis that we have is that if at least you start collecting the data with the high-quality metadata, with good records, and start the process of keeping the metadata up to date and the links and so on, then that enables everything else that people want to build on top of it. And the clear thing is to appeal to self-interest first. And the major thing with the self-interest is the students who, like the rest of us, can't remember what they did 6 months before, and certainly can't remember all the details, and it takes a long time. So if you can actually encourage them to save the time at the beginning, they rapidly begin to learn how to deal with this problem. And especially when confronted with a student they see a student a few years ahead of them who is writing up their thesis, scrabbling around trying to find the original data for a figure because it needs changing. They're quite quick to learn that there are alternative ways of doing these things. And once you've started that process, then sharing with other people becomes much easier. So let me just really stress the major point of usability being one of the most important features here of any software. It need not do everything, but it must be easy and simple to use, and it should really be reliable. But again, it doesn't have to be 100% reliable. We do then want to move this around to actually share the data with some constraints, and this is an extract from The Onion, maybe you've seen it, about the issue about where people are thinking about whether they should or share data, or at least the metadata, and people can have some pretty strange views on this. We have, over the last few years, extensively used the blogging idea, but we built our own sort of blog system because it's a data-centric blog, and we can export material from there that's been used as a collaborative tool in a research group containing about 10 people, and use all the sort of tools that are available to process information that comes from that, to see things like timelines. And that enables the collaboration. And people are very happy to use that. We can analyze the tools to see different ways in which these things are being used. And that management information about how the collaboration is or not working can be reflected back. We also, to support that, because one of the things that happens is you then see a discussion about problems going on. You want to find out why the problems are occurring. And that often reflects back to some issue in the laboratory, something has gone wrong somewhere. So getting the quality environmental data about what is happening in the laboratory, temperatures, working of equipment, and feeding that automatically and being able to associate that with the experiments that are being done, making it much easier to say, this group of experiments was a bit of a problem. Well, the air conditioning was slightly failing at that point. For example, that sort of thing. And automating that process and making it easy to find is something that we're interested in. It does, however, mean that all your results are potentially exposed, at least to your supervisor, and maybe more widely. But I think we should just be much more open about how difficult science is and how frequently experiments go wrong. And it's bad for you if they go wrong all the time, but they will go wrong most of the time. But in the end, you will discover why and make it work. And to quote Jim Gray, and as I understand it, may all the problems be technical, because really most of the problems are actually not technical. They're to do with the way the people are interacting, and whether you need to actually get the people to use other people's software rather than make it themselves. But all the software really needs to be focused on reducing the time needed to look after the data. That's one of the major time-consuming areas in the lab. And the amount of time we spend transforming the table into one form from another, not for a file format, but because for real reasons to visualize the data differently, is huge. But this is no substitute for sitting down and thinking. And this is another, 48-hour internet outage plunges nation into productivity. There are sometimes good reasons for staying away from the computer, and I think we do need to remember that. But what I really want, with all of that, want to say is that the researcher has to have a personal ownership of their information with regard to the whole data life cycle. They have a responsibility to record high-quality data, but if they want people to use it in the future, and since we believe it's good data usually, they should be thinking about how is somebody going to make best use of my data, and with that in mind, how they should actually record it.
Thanks, Jeremy. Okay, now we're going to do a little data switch here. Shouldn't take too long. And next up is George Jorgowski from Caltech. George, take it away.
Thank you. Good afternoon, everybody. I'm an astronomer by training, but I will not be talking about astronomy at all. Instead, I thought I should focus on some of the issues that I see to be of very common interest to all of us. The first thing to contemplate is that the role of computation in science and anywhere else has changed. And in particular, it used to be that computation was regarded as somehow an inferior thing to theory and experiment, the real science that has been with us for several centuries. But this is no longer true. Computation is now becoming necessary and, in fact, may becoming the dominant component of scientific method. And there are two reasons for this. One is that all simple stuff has been done. And if you want to understand complex phenomena, anything in biology, climate, et cetera, you need complex data, and a lot of it. The second part is, I think, even more profound, and that is that we really see now the rise of non-analytical theory, that systems that cannot be solved analytically a priori, and you can prove that, and can be only numerically modeled. So the role of computing has changed from pure number crunching, which has been with us since von Neumann, and we're still doing it, into more of a data and information manipulation. When you use computer nowadays, you very seldom actually solve a lot of PDEs. You usually move information from one place to another. So that's where the data-driven science comes in. Another point is that all science is in fact becoming computational or eScience, and this is a transitional period. In 5 or 10 years, it will make no sense anymore to talk about eScience because that will be the science. So with that, with all the opportunities comes really need for new scientific methodology that adds to the old one, and there are common challenges to all disciplines that we have to all deal with: managing, storing, accessing, discovering large complex data sets, but then we have to extract knowledge from these data. And then finally, something that I think is important is that there is a great virtuous cycle between science and technology applications, that we buy computers in a store to do our science with, but our discoveries will eventually result in new important technologies for everybody. So here is just a grossly oversimplified picture of how, say, modern scientific process works. First, you gather a lot of data from telescopes, gene sequencing machines, environmental sensors, whatever. Then you have to do something proper, housekeeping, store them in right databases, index them, make them interoperable. And we know how to do this, right? Then comes the whole point of data mining or knowledge discovery. And we kind of know how to do this, but not in a very good way. And we still have ways to go. I think there are a lot of technical challenges in that I will not go through. And finally comes what ought to be the really role of a scientist, the creativity and imagination, where you actually interpret and understand what's going on. There are feedback loops all over this diagram, which I didn't write not to clutter things up. So when I thought about what is new and different here, why isn't the same old science with more data, I came up with two things. The first one is that for the first time in history, we will not be able to see all of our data. There is just way too much of it. Up until recently, you could actually see, human can look at every piece of data they measure. We cannot do this anymore. Even in terascale regime, this is hard. In petascale regime, this is simply impossible. So we need technologies that can look after data on our behalf and do it correctly. But then the really important part is that in these complex, massive data sets, there will be features, constructs, important things that the universe is telling us that we simply cannot grasp because they're too complex for us to see at once. And this is where we need some very serious machine help in data mining and knowledge extraction. So this is what I mean by forming a new scientific methodology for the 21st century. These are the skills and the tools that all scientists will need everywhere, and we still don't have them. Well, let me focus in particular on one problem, and that is the data complexity. It's not the data volumes that are important. We talk about petabytes and what to do with them, but just buy more computers. The really difficult part is the complexity of data, which usually comes as dimensionality of some parameter space. And the existing data mining algorithms don't scale very well with dimensions. Clever ones go as n log n, where n is the number of data vectors, but they go some power of the number of dimensions, which is usually unfriendly, and very soon you're in real trouble. Not even the biggest computers will help. An even worse problem is visualization. That there are these structures present in our data which we simply cannot see. We can project them into three-dimensional subspace, but some information is usually lost. How do we do this? How do we actually translate structural information that's present in these complex data sets into something that we can wrap our intuition around without loss of information? This is an enormously difficult problem, and there are no solutions to it. Visualization is important because that's how you bridge from quantitative measurements into your understanding. So a few thoughts about eScience. First of all, it's computational science is not the same thing as computer science. And in some sense, like mathematics is not the same thing as physics. And then within computational science, there is a traditional part, which is solving a lot of differential equations to model something. And there is this new data-driven science. And in fact, the first reduces to the second because the simulations are now generating terabytes and petabytes of data, and their output is also complex, and knowledge has to be extracted from these computer-generated data as opposed to sensor-generated data, and then they have to be matched. But more importantly, I think that data-driven science is not about data. This is a misplaced focus. Our business is to discover knowledge. Data are just incidental to that. So yes, we need to know how to deal with these large and complex data sets, but we're really in knowledge business. Second thing is that most people still think in terms of data poverty regime. Once you have terabytes and petabytes, data are cheap. What is expensive is expertise and skill and creativity, how to extract knowledge from them. And it's not unlike, say, hardware and software situation where computers are becoming asymptotically infinitely powerful and infinitely cheap, but the software is not getting any cheaper. The final thought is that now we're using the applied computer science as new mathematics. Mathematics has served us from the days of Newton as a logical, orderly framework to establish and organize our knowledge about the world and push it further. This is exactly what we're using computation for today. Doesn't mean we're doing it well, but that's exactly what it's doing. So in some sense, computation, in some generic way, is playing the role that mathematics used to play and still does in our quest to understand the world. And just as mathematics served as a universal language of all sciences, able to translate any particular phenomenology into the same logical structures, in the same way, computation can serve as a bridge, as a lubricant, a glue, whatever you want to call it, among different disciplines. And this is an important thing. Here is the usual metaphor of different scientific disciplines, of silos or chimneys, and we suffer a lot from this. This will no longer do, because the important things that we have to do, like sustainability, climate, et cetera, all these things are very intrinsically interdisciplinary, and the academia does not encourage, in fact, tends to discourage genuine interdisciplinarity or cross-disciplinarity. Well, computation, or the methods of computational science, or eScience, are indeed universal substrate, just like mathematics is, which can and should serve as the glue or lubricant of genuine interdisciplinary collaborations. So to summarize, what I think are some of our key needs is we need better scalable algorithms for knowledge extraction from massive complex data sets, and that will certainly include some machine learning, artificial intelligence tools, because we need silicon minds to help our minds comprehend what's in our data. A part of that is we need much, much better way of visualizing highly dimensional data spaces, because this is how you really get to understand as a human what it means to have some 65 covariate correlations and so on. Parts I didn't talk about at all because of the lack of time is what you may call the art and science of scientific software systems, communication and collaboration issues, and the educational issues. All of these are extremely important, I think, cross-cutting and valid for everybody. It's time for academia to do its part of social contract with the society. We need to help develop these things because not only we need them in every field of science, but so does the rest of the modern society and the economy. And these tools and methods are needed everywhere, and I think they're also highly portable or should be portable. Thank you.
Thanks, Greg. Okay, now it's my turn. I have to shift back to the original laptop. Okay, it's plugged in. Okay, thanks. For the last 3 or 4 years, I've been working with biologists on various problems, HIV vaccine design, and looking for genetic causes of disease. And I must have interacted with many dozens to probably 100 different, what I like to call do-it-yourself biologists. And by that I mean, obviously, a lot of science is do-it-yourself, but when it comes to managing data, storing data, analyzing data, sharing data, they do it themselves. They're not working with databases other than perhaps exporting and importing from someone else's database. They're usually doing their data analyses and data entry with spreadsheets like Excel, and occasionally they'll export and do some stats and visualization with certain packages. But they're on their own. They don't have a bioinformaticist working with them. And a typical process that I've seen these folks go through, including myself, I put myself into this category in some respects, is they'll enter data in a spreadsheet, or they'll get it from someone else in ASCII format or a database. Then they'll clean and massage that data into some format that then allows them to run an algorithm. They'll take the output from that algorithm, massage the data again so that they can visualize it. Then they'll finally get to look at the output of the algorithm, and then they'll repeat this process over and over again. And those of you who are do-it-yourself scientists, I think, will sympathize with this in that most of the time you spend in this process is what I've shown here in the large font. It's cleaning and massaging the data into a form that you can use it, and then once you get an answer back from some algorithm, massaging that data into a format where you can visualize it. This takes the vast majority of the time. And, well, what do we do in these situations? Well, we use these very small or atomic data transformations to get our data from one format to another. And spreadsheets, Excel, for example, has a lot of these tools that are very useful. I've listed some of them up there, sort, filter, graph, and so on. But there are some very, very common functions that I've found and I've seen my collaborators come across that you don't have in Excel or any of these common spreadsheet programs. And there are things like joining data, a SQL join, going from what I call dense to sparse, going from what I call sets to normal form. And I thought I'd just show you a couple of these. I think you all know what join is. Here's an example. We've got two tables. Table 1, it's just got an ID and one variable. Table 2, the same ID and a different variable. And you want to join these two tables, so now you get one table where you've got the ID, common ID here, and then variable 1 and variable 2. So for example, ID 1 is associated with A for variable 1 and associated with values alpha and beta for variable 2. So here you have 1A alpha, 1A beta, and so on. Sometimes it's useful to do the join, sometimes it's useful to do the unjoin. And here's another operation, what I call dense to sparse. Often you have data stored in a dense table where each column corresponds to a different variable, but sometimes you want it in this form where the variable name is actually in a column and then the value for that variable is listed in a third column. So the sparse format always has 3 columns, whereas the dense format will have an arbitrary number of columns. And you want it, sometimes you want to work in this form, sometimes you want to work in that form, and you'd like to go back and forth very quickly. And then another very common operation that I see is this sets to normal form. So you might be given data that looks like this, where the variable has a set value, a set of things, but then you want to move it into normal form as shown here. So 1 ABC turns into 1A, 1B, and 1C, and so on. Sometimes you want to work here in this form, sometimes you want to work in that form, and it'd be great to go back and forth. So these tools aren't available in Excel, so Carl, Katie, and I sought to make good on this, and we've built an add-in for Excel 2007, and I'll show you how to install it in a moment, but basically you define your tables, press a button, say to get the join. It'll tell you which columns the two tables share, and then you can select which columns you want to join on, then select inner join, left join, right join, outer join, and so forth. Say go and get the result. So you can download this, either the runtime that you can add into Excel or the source code from CodePlex, and it's available here. So that's all I have to say. So now I'd like to open it up to the panel. I noticed one interesting thing about this panel is we didn't get together ahead of time and say, okay, you cover this, you cover that, you cover this. But it's very interesting that we all have rather different perspectives on what scientists really need. So that's one observation I have. And before we open it to you, I'd like to open it up to the panel amongst ourselves here. Was there anything that you saw that resonated, something that you didn't talk about that resonated with what you saw someone else speak about?
It seems to me that everything everybody said had some resonance.
Yeah, it's amazing we all came up with something different, though,
Yeah.
without trying to coordinate that. Yeah.
I guess one of the things that I would say is I'm interested in the comment about make the open argument, which we could have, and in particular with DNA. So the problem really is highlighted with this. You will only be willing to put up the DNA of a person if it's anonymized. But if it's anonymized, then you don't know where it came from, under what conditions, or anything like that, which means that you've not, in fact, put up the data with the context. And you can't afford to put up the context because you need remarkably little data to actually narrow down to the person. But of course, there's a lot of very valuable information in understanding where that person was, whether it's a racial issue or with the epigenetic stuff, the history, that person, and so on. And actually, there's a lot of very useful information that come out of it. We have started tagging some of our data with some geospatial information, even though it really actually is not obviously important, but it's very easy to add. So we record which room it was in, which turns out to be quite useful when we're trying to remember whether we used a glass dish or a Teflon dish, because we should have recorded the correct information, which was what dish. But in place of that, we recorded some other information which helps us work out what things went on. But, I mean, that's a slight triviality. But it does show that in many cases, there are constraints on what context you can make available. And so whilst I'm for making context available as much as possible, I think it has to be realized that sometimes you will have to start again. and you will have to repeat everything. And in science, it is perfectly permissible to repeat stuff if you can, but you don't want to do it if you don't have to. There ought to be a reason. And having forgotten where you put the data is not a good reason. Finding out that there was a problem with the original experiment and you would like to do a slightly different one, or repeat it to see if indeed whether you get the same results because you've got reason to believe maybe that that hasn't been tested, is a perfectly valid reason for doing things.
Well, one thing that struck me is that there is an underlying issue that we kind of touched upon, and that's the issue of usability. Scientists are as intellectually lazy as anybody else, and they're really reluctant to learn new stuff unless they absolutely must and have to. And sometimes they don't know that they have to. So making less steep learning curves for these tools is a very important thing. Scientists tend to respond to positive stimuli of two kinds. One is discovery and the other one is resources. And if you manage to create some exciting, genuine new results, people respond to that, say, gee, I want to do that too, so I'll go and learn. And of course, the resources that we're not allowed to talk about. But there is some strategic thinking to be done about that. And it's an unglamorous thing to write beginner's manuals and worked out examples and do hand-holding. But if you really want to spread the effectiveness of these tools, you have to invest in that somehow.
I would strengthen that some more. The last slide I didn't show actually has, don't forget the users amongst all the semantics. The semantics are extremely good at making computers understand each other, but they're not much good at making humans in many ways understand it. And it's not just the manuals. In many ways, the user interfaces is a hugely important part, and the easier these are to deal with, the less you will need the manuals. But you have to remember that in the global collaborative arena that we're in, cultural differences are huge. I got sent a PowerPoint file, which I looked at on my tablet, which was sent from Israel, from some friends, and I suddenly realized that the arrows for going forward and backwards were on the right-hand side of the PowerPoint. It being the tablet, it's just the screen rather than the left-hand side. And that's fine. I perfectly expected that. I can read right to left as well as left to right. But the problem was I didn't know which one of the two arrows was forward and backwards. The context has changed. There are many languages that are right to left, not left to right, and it makes a whole difference as to the way that you view what the screen in front of you is presented. But most of the software is designed for one particular cultural experience. And even if you come from one discipline to another and then look at the same piece of software, many of you will also have the experience of trying to persuade your partner how to approach some piece of email software or something like that. And in the case of my wife, has a perfectly valid reason for expecting something to happen when she hits one of the buttons. It may be right, it may be wrong, but it's certainly very different from the reasoning that I would have gone by. So I think there are all sorts of reasons why people will approach an interface in a different way. And we need to think about that, as well as providing the manual, but actually also think much more carefully about what is presented on the screen and what people will think about that.
All right, so now's your chance. What do scientists really need? Or is there something that you've heard up here that you would really like to disagree or agree with? Tony.
I don't know if the microphone's working.
Yes, it's working.
I'm all for openness in data and metadata and so on, which you've all sort of in various ways addressed. But, Jean-Claude Bradley has an open chemistry blog, and it seems that they put up data which has hardly been digested, right? So it's something that a research group does, and there's errors in it the students have. It's fine, that seems to me, doing that privately, but would you want to make all of that data public? Surely there's value in doing the sort of academic thing and sorting it out before you make it public. Isn't there a balance to be struck there?
I personally think so, yes. And I don't do the equivalent of John Claude Bradley. We make that data available in a research group, but I wouldn't want to inflict it on others. However, if and when we have had a reasoned argument and we make the high-level stuff available without the underlying material necessarily in your face. It certainly, there is value for somebody being able to drill down through that so that they can check what has happened. But if everything holds together, and until it appears not to, there's no need. And I think Jean-Claude Bradley does an extreme of open notebook science. I don't think there is much point in doing that personally for everybody. I think as a demonstrator it's fantastic. But we've been having discussions, those of us who were another Microsoft meeting, about the people in the group who did or did not put stuff on Facebook and what the value of the material that was on there. I think there's an equivalent here. There is tremendously valuable information can be transmitted around, and there's information that should be available if needed, but doesn't necessarily to be up there in front of you and potentially confusing.
Right. There's a difference between making it possible and making it easy.
Yeah.
I would tend to take an opposite view with respect to that, but perhaps DNA and genetics is slightly different from chemistry. When one runs a DNA sample on a SNP chip where you get genotyping information for maybe 555,000 SNPs or maybe even 1 million SNPs, that raw data is very valuable to the next person coming down the road who wants to do a genome-wide association study, because what he can do is he or she can do is recognize that this data is digital, download it, add it into their own data set, and therefore increase their numbers of samples and increase their the power of their study. So I think that in some contexts there is value for putting the raw data on. I think that what you may have been referring to is this idea that you're putting raw data on that may be incorrect. I think that you'd want to try and avoid that. You want to put the data on when you know it's reached a certain level and you're confident in its accuracy. But once you have reached that level, the day of publication and sometimes even the day before, we try and dump everything onto the web.
Yeah. So I run a micrometeorological station, and where we measure all the things that we need to solve the surface energy balance equation. And we had a policy of going through a data quality check before we published it. And that just turned out to be a choke point, because we didn't actually catch all the errors, and it significantly delayed getting the stuff available because whoever was in charge of the data quality check had other things to do. And so we simply put our data out, but we indicate what's been gone through a quality analysis. But there is this problem of a choke point, because most of us don't have someone who only does data quality checking as his or her sole job.
Yeah, I mean, I think that it comes back to the point that I think people need to consider what the value of the data is at all its different stages through a potential life cycle ahead of it. And if you can see that there is value in having the data, even though it may be error-prone, or whatever reason, I mean, it's sometimes really very helpful to have the rawest, sometimes most useless data out there, at least in a few cases, as examples, which are very, very hard to find if you're trying to teach. It's not there. But it doesn't mean everybody has to do it. Sometimes, though, if everybody's doing something, it doesn't matter that it's error-prone, because actually you can broach the whole system in a different way and use a statistical analysis on the whole set. If there are thousands of your micrometeorological stations out there, it's very unlikely that they'll all be in error in the same way. And so this mass citizen science sort of thing, then that works in a slightly different way. So I think it very much depends on what the data is, what the potential issues are with it. But raw data, I certainly, prior to publication, raw data should be available. Sometimes even the negative experiments, the ones that failed, can be very useful. But I think you do have to consider it very carefully. And there are extremes here. But ultimately, results that are made public, I certainly strongly believe, have to have their context, and the context is the raw data.
Great, Kat, I think you're next.
Sure. This is mostly for George. So I fully agree that there are great challenges in data-driven science, and that a lot of it has to do with the complexity of the data. But I think there are equally important challenges for the complexity of models. Computation is not replacing model-driven science, I don't think. I think that we're having more data, and our models are becoming more complex. We've got scaling ability now on both fronts, so that we can have richer data sets and richer models. And there are fields like ecology and biology where, relative to the complexity of the models, we still have a great paucity of data, and that we need to do lots of computational work for scaling up to the model complexity too. And my view is that that the fundamental nature of science, the balance between models and data, has not changed. They've both gotten more complex. I just want to see if you agree with that, or if you really think that it is just data-driven science.
Well, I didn't mean to imply by any means it's just data-driven science. I completely agree with you. I was trying to say is that computational science, be it numerical modeling or data-driven, is becoming increasingly more important component of the scientific method, which a lot of people do not fully appreciate yet, and somehow consider it intellectually inferior part of scientific inquiry.
No. Well, you don't have to convince me that that's an issue. But the traditional computational modeling, the kind of first principles, where you have a few equations, you do it many times, I think that's under control, but in a lot of ways. But what's not under control is models that have many components, like large ecological models, large regulatory networks. Right. That's a whole different class of issues that parallel the data-driven challenges.
Yeah, no, I agree. I simply didn't have enough time in 7 minutes to talk about everything.
No problem then.
Peter Murray-Rust, I'd like to ask about graphics. I've been doing graphics for 30 years, and we still don't seem to be able to deal with two-dimensional graphics, let alone multi-dimensional graphics. We do not have a web standard where you can put up something which is a semantic graph. We had scalable vector graphics, which I thought was going to help us, and it's been trashed, and we now have Flash. So we actually have a position where we cannot transmit data except to sighted humans. We can't transmit it to machines, or to humans who can't visualize it. And really this is aimed at Microsoft people particularly. Do you think there is a way where we can actually assume a common graphics environment on every machine in the foreseeable future, where you pass a two-dimensional graph, a plot of x against y, and you don't have to think about plotting it and rendering it and being able to deal with it? Or are we still going to be stuck in this mess of different formats?
Anyone in the audience? I am not an expert in that area. Nope. Okay.
Yeah. But it isn't just a graphics problem.
Well, we do have graphics standards, but just too many of them.
We don't have graphics in the operating system.
Okay.
So Apple had it 20 years ago.
Yeah. So one of the things I've done in my own graphics work that has proved to be very helpful in addressing this problem that Jeremy mentioned is that I do most of my graphics in MATLAB. But what I do is when I make a graph, I try to save a little .m file that makes the graph for me. And so I iterate until I get something that, in fact, produces the graph by just typing the command. And, when the paper comes back and I need to revise something, or years later I want to try to recreate that graph, it turns out to be a very helpful strategy. I can go right back to it.
Until your program becomes obsolete and no longer works.
But you still have the code is written in is readable code.
Yeah,
the equivalent that we were just talking about, R, because you can run R under this Sweave system. So either with HTML or with TeX, it is actually possible to write this document that reproduces itself, and the code is in there, and it can print it out as well. So there's a very interesting statement if you look at some of the history of Sweave. It says, so anybody who has access to the data can reproduce the document. And that is a very big if, because within reasonable computation you can do it, but these reproducible document standards haven't really gone down the line of, so where did the data come from and how do you place it somewhere? And a little bit extra work there, which we've certainly been thinking about. So if it's a not too big a file, you can place that with the URI somewhere. The R documents, MATLAB will do it as well. Various others will read it in from a URI. And there you've got it. Now you really do have a reproducible document. They've got the original data and they've got the file. But of course there are all sorts of limitations on that of computation and scale and size. But at least it goes some way to transporting a bit more information. At the moment all we can do is to send the table. of data with the graph, which at least means that you've got something to work on. That's something that is not always done, but it's a minimum necessary step to really transmit the information. Of course, if the graph has millions and millions of points on it, that can be quite expensive.
Okay, over here.
Okay, so Luke Church. I'm a usability researcher, so I'm very glad to hear of the emphasis that was placed on it. And my question is essentially, what does science need from the kind of things that human-computer interaction might be able to offer in order to make progress with the usability of tools? So other disciplines, for example, have needed advocacy or training or cheap analytical tools that designers can use or evaluation methodologies. What would work best for you?
Our limited experience in this area, some people may know about our studies on making cups of tea, and a work by analogy, so that if you want to create an electronic lab notebook, which was just one part of what we're doing, that will work really well in a lab, so actually with you in a lab, recording the information you really need to do it and deal with whatever the issues are, it's very difficult to take a computer scientist, even former chemists, of which there are a large number who turn into computer scientists, into the lab. They're usually rather dangerous hanging around in the lab. And they find it difficult to see through lab specs and so on. But if you can find something that has got most of the issues that will come up, but is safe and easy to use, and is in principle something that both parties understand, and the complexity is making a cup of tea, which defeats most parts of the US, I have to say. So it's not straightforward. Boiling water is necessary. It's not an option, not warm water. But anyway, but that is exactly, by having something like that everybody can understand, gets you quite a long way. But I think thereafter the whole issues of doing these things by, I used to think this role play stuff was a bit of a joke. I still don't like doing it. But I have realized that actually if you get into it, you don't actually need the chemistry set there, you don't need the computer there. The sketching on the piece of paper works really quite well. And you really immediately sit there, I was watching the student doing the experiment and recording that way, and then thinking, there's no way they will actually do that on the computer. Okay, so the case is one, the role play worked. I could, without doing it, say that is not an approach that will actually work in the lab. And I think we just have to get more engaged, which means more time being spent on it.
Of course, being the Irish person, I have to say that the most important thing about making the cup of tea is whether you use Irish breakfast tea or English breakfast tea.
Which whiskey for that?
And the time of day, yeah.
So Jeremy mentioned it during his presentation, but when we go back to the question about what scientists really need and the talk about standards, so Jeremy's work in chemistry, where he talks about maintaining the protocols that go on in his lab and, for example, tea, are the protocols that he uses able to be easily translated, for example, into a lab in, say, for example, India or China? Would they just be able to be picked up immediately and used? Because the data that Jeremy generates may be very useful and may be able to be reused, but without the protocol that actually generated it in the first place, you actually start to lose, once again, as he said, the context. And for example, where Brian was talking about the SNP chips and the DNA, do SNP chips that were built here operate and provide the same output information or data as those that are built in, say, for example, China, India, elsewhere? Are they using common formats and things like that? And then we hear about things, for example, documentation that's stored in formats that can no longer be opened, no longer be used. And from that point of view, we need to be thinking much more long-term. We think at the moment very much in terms both within our professions and within society in short-termism in reality. We need to be thinking beyond 10, 20, 50 years from now, where this information, if we're going to keep it, will still actually be of value. And what does the panel think about that?
So I think that with respect to SNP chips, I know this is really your more gestalt question, but just specifically about the SNP chips, they are manufactured by a small number of manufacturers. It's the same protocol you're supposed to use no matter where you are in the world. When I look at data from generated in England versus China versus in our own lab, I know the quality control checks that I need to look at. And it's a very important aspect of when we want to unite data sets across labs, meta-analysis wise. But I think your point is very well taken. I think that is a specific example where there's a very clear protocol and it's very well worked out. There are many other instances where it's not so well worked out and it's not so clear how you can sort of step between labs, as we call it. So I think it's a very important point. I think we struggle with this on a day-to-day basis. There's no easy answer.
I think there are some things that are just best learned through apprenticeships. That is, you can write things down, but it's pretty hard to learn how to cook with just a book.
I mean, in the end, the analytical chemists have this problem. You have a new piece of equipment, a new technique, and it has to be validated. And validation is a very serious issue across the whole. It used to be, the days when you could get a small group of people together, they could see all the data being generated on a particular type of approach, they knew all the labs or they could visit them, and they could actually give you the considered opinion. That's gone. It's too expensive, it's too difficult. So issues of quality control checks and providing that information is necessary. But even so, one of the things that happens in the EU, I think it happens worldwide in some other cases, is they will get all the manufacturers together, or all the labs who will do some experiment, and may actually physically bring the equipment to the same place to make the ultimate tests. Now, that's with pieces of equipment for use right now. And if they have to go to that extreme for that sort of test, then we have an inordinate problem over the long term. So I think we just have to work out how to provide as many potential checks, as much information as possible. But in the end, there's a continuity, I'd agree, there's a continuity of information that is learnt by passing on information. And it's very difficult to write that down. Whenever you try and tell somebody to do something and write it down, you often end up wandering around to their office and showing them what to do, because you forgot something that was obvious to you. Hit that key there. It's very difficult. And with the usability side, if you really want to, you have to have somebody watching you who is trained in writing stuff down. And that's a very expensive procedure to do. But if it's critical, then that may be necessary.
Actually, just to come back to your point, it's interesting. DNA labs, genetics labs are evolving along two pathways at the present time. There are actually genetics labs who never do any experiments at all. They send all of their DNA out to core facilities that have established quality control checks and protocols, and then all they get back is the data. They give the DNA plus money, of course, and they get back the data, and then they specialize in analyzing the data. In my lab, we have tended to stay away from that for a variety of reasons. So we like to have the machine on site, mainly because we like to maintain our own quality control checks, but also because when you get a new piece of equipment, for example, the Celexa, which allows sequencing of a tremendous amount of DNA, much more than was previously available, there's 1,000,001 experiments that you can think of, crazy things you can do with the machine that you hope nobody else has come up with. It's not going to be in next week's Nature Genetics. But if you are giving them out, if you have to send your samples to a core facility, that's not going to happen. It's better to have them in-house.
Jeremy Walton. I was very interested in George's point about the use of visualization as a way of, as a standard way of interpreting data and presenting it to scientists so they could better understand it. And I thought he made a very good point. As these data sets become much larger, most of the time we're never going to see all the data. We're going to see some sort of summary of it as presented, for example, in a visualization. And I was interested in his point about the difficulty of multidimensional visualization. I wonder if he could perhaps expand on that a little bit. I was wondering if it's just a really hard problem to do because nobody's really thought of a good way of doing it, or that there are various different techniques of doing multidimensional visualization, and yet, if we were presented with a visualization of 13 variables or something like that, would we really be able to understand it, even if somebody came up with a good technique? Are there problems of cognition, of perception, of all these other things? Leaving aside Peter's point about whether or not we could ever get it from one web browser to another one.
The short answer is yes, but now let me expand a little. there are techniques for multidimensional visualization, and I can think of ways to encode up to maybe a dozen dimensions into a pseudo 3D display. But you kind of tend to get a headache after about 5 or 6.
Right.
And on the other hand, you don't need to perceive the full 12-dimensional scene, but you may be able to perceive outliers or anomalies or unusual things that stand out in some subset of those, and in that sense that works. But I think that's just about it. And I'm not an expert on this, but from what I can gather, that's roughly kind of number of dimensions that people know how to encode in some graphical fashion or other. But that doesn't help, because in astronomy I know that we're now dealing with 1,000-dimensional data sets. And we're all used to look at X versus Y plots on paper, or now maybe X, Y, Z, and there are correlations and clouds and clusters and what have you. But there could be, correlations in some arbitrary subsets of those dimensions, clouds and clusters that have weird topologies and so on. And you mathematically know what that means, but you cannot visualize it, right? You cannot establish intuitive connection between some patterns. Because in some sense, what science is all about is really reducing the messy observed complexity of the world to some simpler subset of rules and laws or whatever. And if you can't even see the messy complexity of the world because it's intrinsically present in 1,000 dimensions and it does not project well onto any 3D subset, well, then you have a real problem. So I think this is a very serious research problem. And as far as I can tell, there isn't a heck of a lot of work going on it. A lot of visualization research, my impression is, it's entirely driven by the entertainment industry making better blood and gore graphics for video games. So better 3D visualization all the time. Fantastic 3D visualization. 300-dimensional visualization, you're out of luck. So until somebody thinks how to make money of it by looking at 1,000-dimensional stock market parameter space, right?
There's no idea. Thank you.
And when visualization fails, you turn to things like machine learning, and the computers start looking for patterns, and they do the best they possibly can.
I'm all for having smart robots telling me what to look at.
Right. Okay, one last question.
So I guess my question's a little bit different. So we've been talking a lot about data, and once you know what experiment to run, then you're going to generate data for genes or the astronomy or whatever sort of domain you're in. But I guess when I read this question, I thought, what does it take students to, in the doctoral program, where is it that students sort of seem to falter a lot before they can really sort of get into the dissertation and graduate? And the biggest time frame is in formulating the research question. So, I mean, once they've got their question, then it's sort of crunching the data and they struggle with all the issues that we've talked about. But I guess my question to you is, if we go before you actually generate the data, how you actually come up with your research question, do you see much hope for sort of systems that might help with serendipity or creativity, or is that just something that's inherently human that systems are not going to be able to help us with? Or maybe there's no creativity in science. That's, I guess, another solution.
Well, you could encode serendipity, like unsupervised classification methods. You can ask proper algorithm to go and discover things that are unusual in some statistically defined sense. So you can certainly have machine-assisted serendipitous discovery like that. Creativity, on the other hand, is a little too nebulous concept, I'd say. Well, there is something for cloud computing.
So Ralph Keeling had a really interesting essay in Science in March. Ralph is the son of David Keeling, who was the scientist who started the carbon dioxide measurements 50 years ago at Mauna Loa and at the South Pole. And, that turns out to be one of the most valuable data sets that we now have in science. And in some communities, you can put a graph on the screen with no labels and no numbers on any of the axes, and most of the audience will immediately recognize what's going on there. And so on the 50th anniversary, his son wrote an interesting essay, and what he pointed out was that the motivation to measure CO2 on Mauna Loa was not hypothesis-driven. And so I think in some ways there's, we may be doing students a disservice with this sort of logical positivist view of the way science works. And you certainly see this when you, say, are a judge at a science fair, is that nobody kind of goes and makes observations on the basis of curiosity, and at least in science fairs. And I think maybe encourage people to do a little more of that.
I suppose in genetics I'm a little bit more fortunate because we're now data-driven, not hypothesis-driven. I don't care what the hypothesis is in genome-wide association. In fact, the whole point behind genome-wide association is that there is no biological hypothesis. I'm testing every gene and every portion of the genome with equal stringency, which moves away from the forerunner, which was known as candidate gene approach, which is basically where you pick a gene based on what you thought was the biology of the gene and the biology of the disease. But we know from previous studies, for example, there's a database where somebody has collected, Rudy Tanzi has collected all of the published candidate gene papers for schizophrenia. And there's something like 3,051 variants that have actually been published in sometimes in very top-line journals, stating that these have an association with schizophrenia. What he's done is he's gone and he's looked at all of the subsequent data that's been published on these variants. And out of those 3,051, four variants were actually of sufficient quality to be considered true association. So this idea of candidate gene approach really is dying, certainly at the present time, with the advent of genome-wide association, which tests everything. So we move away from the hypothesis driven, which is a problem, I will tell you, when it comes to writing grants, because reviewers go, well, it's not hypothesis driven. Well, it's not supposed to be hypothesis driven, it's data driven.
So just to finish off, I think the general overall hypothesis, why I want to investigate some particular area, that's perhaps a bit tricky. But there is a level that once you've decided that you're going to go down a particular track, or the funding is for students to investigate some particular thing, and they collect some data, the biggest problem we then have is getting them to think about it before doing the next experiment. Actually think about it, discuss it, start the interpretation, therefore be more informed when you do the next bit. Do I need more points at this end or that end, or in the middle? And so the sort of blogging stuff that I showed you where these discussions are going on, it's a data-centric blog because you're discussing data almost always, much more commonly than even the literature that you may be discussing around it. But it's encouraging that discussion. And that's the thought process that is needed. And one of the biggest delights I've had by using this, I've used it in a group that I run with about 10 people. And that's mainly used for not the rawest data, it's just discussing stuff and so on. And that works very well. And changed the way the group meetings run. You no longer have to specifically tell people to prepare material for discussion in the group meeting because it's there. You just bring up the blog page. And if you don't put anything up that week, you're not going to get discussed, or only towards the end. So that works extremely well. Reduced the barrier and got the discussions going. In another part of my group, the more chemistry side, where people are doing more individual projects, one of the things I now get is I walk in the morning and one of my students may say to me or send me an email, have you looked at the data that I've put up on the blog? It's not, can I see your data? It's, have you looked at it? This is what I think is going on. Here is the summary of it. That's a very interesting change in balance, and I think extremely effective at promoting the responsibility, again, of the researcher to be thinking about their data and what they're doing with it. So maybe the initial discussion that can be recorded about why you're doing something, that's a bit hard. But the slightly next thing about why you're doing something right now and what should you do next, that discussion that parallels the data and the experiment is extremely important to record and collaborate with. And the techniques we have work very well for that.
All right, let's thank the panelists and thank yourself.